Welcome to Tiny Show and Tell Us, the bonus series where you write in with your favorite science news or factoid. We read your email aloud and then dive deeper. I'm Sam Jones, and I'm here, as always, with my co-host, Deboki Chakravarti.
Hi, Sam. So last time we did this, we talked about insect vibrations. We also talked about long-standing grudges against bad science teachers.
Important.
Yes, very important. And of course, a reminder for all of you listeners that if you have a story you'd like for us to cover on Tiny Show and Tell Us, all you have to do is write us an email. So you can email us at tinymatters@acs.org or fill out the form linked in the episode description.
All right, cool. Deboki, I am going to just go first today.
Yeah,
how does that sound?
Go for it.
So I'm going to start with something a little bit different because I'm going to start with an email. There's no science to unpack in this first little thing I'm going to share.
Got it.
But I got an email from listener Cecilia, and I was out when I got this email. So I was on maternity leave. A lot of people don't know that because we were publishing episodes, but we had pre-recorded a lot of material.
I thought you were about to say we had a baby.
Oh, we collectively had a baby.
Yes. All of you also all have a baby now.
Yeah. So if you could send, money, diapers, food.
Yes.
Yeah. So Cecilia got in touch with us, I think like a week after I gave birth. So I was not monitoring the Tiny Matters inbox. And then I just emailed her back a few days ago. She forgave me for my tardiness.
Very kind.
And then also said it was fine to share this email. So I'm going to hop into it. And Cecilia writes, "Hello, I've been catching up on the Tiny Matters podcast, binging many of the episodes I missed, and wanted to send an email with some info I thought you might find interesting. I'm the listener who shared the Dogs Love You proven by MRI on Show and Tell Us number 6. This May, I graduated with my bachelor's degree in chemistry, fittingly right beside my service dog. Having a service dog in chemistry is something I had never heard of before prior to getting one. The college president awarded my service dog with a special patch in recognition of her work, as my service dog was trained to be in the lab with me. Over the years, my service dog has attended many labs and also aided me throughout my undergraduate thesis research in inorganic chemistry, so it felt like a great achievement.
Yeah.
However, unexpectedly, over 2.6 million others also agreed. On my Instagram account, which is @salad_and_crouton, we went internet viral and ended up being featured on ABC World News, Good Morning America, Fox News, and People.com, along with many other news outlets.
Oh,
incredible.
Wowie.
I know. And then Cecilia continues, ACS has been a huge part of my growth throughout the years and has one of the best communities I've seen. Inspired by this community since 2022, I've been presenting at various events and conferences, advocating for myself and others on the topic of service dogs, invisible disabilities, and navigating STEM careers with a service dog. So I thought it would be interesting to share this news with Tiny Matters, especially as I know the hosts are big dog lovers. I know, Deboki, you have a cat, but you say you like dogs.
I love dogs. They're great.
We know I'm like pretty obsessed with dogs.
And Cecilia's dog sounds brilliant.
Yes. Brilliant, adorable,
Yeah.
all of the things. And so then Cecilia closes out by saying, "Thank you for your time in reading this all over the place email." It really was not, Cecilia. Don't worry. Attached below will be the original video link and the ABC video that started it all. Have a wonderful day, Cecilia Hernandez. So you can follow Cecilia on Instagram and YouTube at salad_and_crouton. We've linked to the Instagram reel and the ABC video that Cecilia mentions in the show notes. And they are worth the watch. And I was mentioning to Deboki right before we started recording that I saw this go viral back many, many, many months ago. Had no idea that Cecilia had then written into us after it went viral. So it's like 6 months later, I'm getting caught up on everything. But this is one of those things that went viral, but is worth revisiting because it is heartwarming. And oh my gosh.
And congrats
cute
on
dog.
your degree and everything too.
Yeah, congratulations. That's super exciting. I just loved that. I just loved it so much. So that is a different way to start a Tiny Show and Tell Us, but I thought it was just so wonderful and it was sent in so long ago and I was just fully unaware.
Yeah.
So now we are getting into a Tiny Show and Tell Us. We're going to break down the science. This one is from listener Binky. I say Binky. It's my sister. Her name is Caroline. I've talked about her on many episodes, but this is from my sister. And this is what she wrote. So she wrote in saying, Hi, Tiny Matters baddies. Baby sis here. I've been waiting and waiting and waiting, in all caps, to have a science fact for you all, but alas, I'm not a science gal, so it took a second. This past weekend, I was out with a gal pal who told me she was pregnant. Yay!
Congrats.
Yeah, congrats. We then proceeded to talk about pregnancy tests and the accuracy between different brands, types, and how expensive they are. At the end of this conversation, my friend said, well, it's better than killing a bunny. To which I said, WTF. She then proceeded to tell me how her mom told her that in our grandmother's generation, they would inject a woman's pee into a bunny and, quote, if it died, then she was pregnant. I did a little digging and LOL, that's not quite it. And then Binky puts in quotes, I'll let you science chicks explain. But honestly, it's kind of worse because they would kill every single bunny, pregnant or not, to find out if you were in fact pregs by dissecting them to see if their ovaries were enlarged. Wow, terrible. Thank goodness for ever-evolving scientific research and practices for both me and my fondness of bunnies and the bunnies on a whole. Thanks for doing what you do and entertaining me for 100 plus episodes. You're welcome, Caroline. So let's hop into it. Let's get into this one. We're going to start with the now. Home pregnancy tests used today have been around since the late 80s, early 90s to detect the hormone human chorionic gonadotropin, or HCG, in urine around 6 to 10 days post-conception, which is like 3 to 4 weeks after the start of your last menstrual cycle. It's a lot of ballparking. But generally speaking, that's when it can be detected. So it's produced mainly by the placenta during pregnancy, which just goes to show you how early placental cells are making things happen. Mm-hmm. You want to hear more about the placenta? Go listen to our placenta episode.
Yes.
So HCG rises quite fast once a fertilized egg implants in the uterus. Home test strips, they're strips that have antibodies on them that HCG will bind to, really similar to like a COVID test or a flu test, like any of those like ones where you get a nose swab or whatever, it's kind of that. Mm-hmm. But urine is used instead. So if you get that colored line, that's the antibodies binding to HCG and then actually binding to another bit of like an antibody strip. It doesn't, the main thing is if you have HCG and it binds to the antibodies, you're going to get that colored line.
Right.
Or you're going to get a test that says pregnant or not pregnant, but that's just a digital readout of the chemical reaction that's happening on the strip.
Right.
Just takes even that tiny bit of guesswork out of it for you.
Yeah, which like, especially if you're like testing like super early and you're like, I think I'm pregnant, but you're not sure. And then you're like looking at that strip and it's like a total optical illusion sometimes where you're like, if I put it in the light this way, it looks like it's positive. But if it's this way, it doesn't.
Yeah. It's kind of, I mean, very similar to a COVID test where you're like, is there a faint line? Do I have COVID?
Yep.
So yeah. And then of course, to detect pregnancy earlier, you can have a doctor do a blood test. That is by far more accurate. You can detect the concentration of HCG in the blood, which will increase exponentially at the beginning of pregnancy.
Yeah, and so that's like also really important for tracking. Like if you have like a healthy, viable pregnancy going on early on, like they'll use the blood test for that.
Right. So you're seeing like typically I think it's an exponential growth curve. Like you're seeing
like it's supposed
doubling.
to double every day, I think.
Yeah. And so, you'll go in and then they want you to come back a few days later to double check. So what happened before the late 80s, 90s when we had this available? Well, in the 70s, the FDA approved what the Association for Diagnostics and Laboratory Medicine refers to as, quote, mini chemistry sets that required users to mix urine with solutions in test tubes and wait 2 hours for a result.
2 hours.
Yes.
Like even waiting 2 minutes is agonizing.
Yeah, 2 hours. So these were kind of annoying to use, which by the description is clear that it would be annoying. And apparently they had a false negative rate of around 20%, which is quite high.
Yeah.
And so I found an incredible article in The Conversation about the history of pregnancy tests written by Helen King, who is a PhD historian of medicine and specializes in the history of obstetrics and gynecology. And she also wrote a book that came out actually in 2025 called Immaculate Forms, Uncovering the History of Women's Bodies. That sounds really interesting. So before we get to the bunnies specifically, let's talk urine as it relates to pregnancy. 3 ancient Egyptian papyri, like I guess the plural of papyrus, which I hadn't ever known before.
Yeah, is it papyri?
Show that urine was being used as long as 4,500 years ago to determine if a woman was pregnant. And I'm putting determine in air quotes. So in these texts, a woman is described who either wants to know if she'll conceive or if she's pregnant. Really different things, by urinating on wheat and barley over the course of several days. And then they say if the barley sprouts first, it's a boy. If the wheat grows first, it's a girl. But if no seeds sprout, she isn't pregnant. Which is like, okay. So yeah, it sounds a little wonky. But thousands of years later, in the 1930s, people were reevaluating these seed tests. And they found that 70% of the time, a pregnant person's urine did make the seeds grow. Again, like it's more than 50-50.
Yeah.
But still it's only like it's 70%. So this is not foolproof. It's not a great error rate, but also the whole like also the if barley sprouts, it's a boy, wheat sprouts, it's a girl. Like, of course, that has no merit whatsoever.
Yeah.
But the idea that there was something detectable in a pregnant person's urine was legit. Like there's something going on. And so around the same time, here's where you get into the bunnies, Caroline, sorry. But also you asked for this explainer, so that's on you. Rabbits were being injected with a pregnant person's urine and then killed to see how their ovaries changed. So the researchers doing this would look for larger ovaries and the corpus luteum, which is the endocrine gland that's made up of a yellow mass of cells and lipids that forms in the ovary after ovulation. But of course, to do this check, the rabbits had to be killed. Later, live toads were used instead of rabbits and injected with urine. And if a person was pregnant, then the female toads would release eggs. So the good news was that these toads weren't typically killed. But the bad news is I found out that their mass export may have spread a deadly fungal disease to wild amphibian populations across the globe.
Excellent.
So, because when I first read about that transition to toads, I was thinking, great. Like is it great for them to live in a lab where they're injected with urine? No, but they're not you don't need quite as many of them. You're not like cycling through them the way that you would if you had to kill them every single time. But then I found out about the whole deadly fungal disease thing. And I don't know. I'm like, what's better? I'm not sure. So none of these tests were great or ethical. But thankfully, antibody work, particularly in the 1960s, led to what we now have available today, which is a highly accurate at-home test that does not use animals. And so, Binky, I hope that you are feeling informed. And
also grateful to have not had to be pregnant at a time of rabbit tests,
toad tests, or having to essentially use a mini chemistry set and then wait for 2 hours.
Or having to go pee out in a field.
Go
so
that's
get
that
like
barley.
Of all the ones we mentioned, that's the one that I'm like, you know what, that one I can handle. Like, I don't like the idea of having to wait to see if the seeds are going to grow. Like by that point, I mean, I guess like back then you wouldn't like.
It sounds like they had to go back multiple times. So you just like keep going back and peeing in the same spot day after day.
It almost feels like by the time you get the results from that test, like you'll start to feel that you're pregnant. Like you'll start to potentially have started to show and have more signs. But it was also a very different time. Like you couldn't ultrasound.
I mean, 4,500 years ago, not a lot of options. I guess I was reading also that I believe it was ancient Greece, there was something where it was like, as soon as you've conceived, you can feel your womb close. And it was like, what? No. Like there have been so many things also then related to blood or urine, or if you like bleed on this type of cloth and then wash it at this type of day. Like there's so many things, but I thought that it was really interesting that there has been this like urine thread for thousands of years. And that although peeing on barley or wheat is not an accurate way to see if you're pregnant, it was just interesting that people were already thinking there must be something in urine specifically that is an indicator of pregnancy. And I thought that was cool.
For sure. Well, Sam, I have a story for you from listener Madison. She sent us a link to this study and wrote, "Ever since I heard about this study, I could not get it out of my head. It's an excellent story about false positives and accounting for natural variations in data. It is also an excellent mental image, a dead fish lighting up an MRI machine. I would love to hear about how this is being implemented today and the lasting impact it might have had. Love the podcast. So
I remember this.
Yeah.
I also, when I saw that someone wrote in with this, I remember also seeing this and thinking, this is fascinating. And I'm so glad we're talking about it.
Yeah. So for those of you who are like me, who actually didn't know about this, Madison's link is to the story from improbable.com. And it's actually about a recent study looking about the reliability of functional MRI to find biomarkers of disease. But it's also about this older study, which is where the dead fish lighting up on an MRI machine comes up. And so we're going to get into both of these. So some background information. Functional MRI looks at blood flow, which scientists often use as a proxy for brain activity. You know, like the idea is that if you have neurons in a part of your brain that are being more active, then there will be more blood flow going on. And so something you might do in an experiment is have your subject do some kind of task and use the MRI to look at their brain and see where there's activity going on. So there's this more recent study that the news article is about, which gets into how reliable fMRI is for that. And I'll get into that in a bit. But we're going to go first to the dead fish lighting up the MRI machine, which the article talks about too, and I want to get into because I didn't really know that much about this. I guess I didn't really do much brain stuff, so this was newer for me. Yeah, so in 2012, the Ig Nobel Prize for Neuroscience was given to a group of researchers who bought some dead Atlantic salmon from the store and put it into an fMRI machine. They showed the salmon pictures of people doing different social things. They asked the salmon to figure out what emotion the person in the picture was possibly experiencing. And again, when I say all of this, I emphasize that the salmon was dead. So you really should not be expecting any brain activity, right?
Right.
But the fMRI data showed that the dead salmon had brain activity. Like it was like thinking about the pictures. So this was all just like a big what? Like they should not be showing this.
Yeah. And also I will say also just a side note on the Ig Nobel Prize is that it's a prize that I think on their website says it honors achievements that first make people laugh and then make them think.
Yeah.
And it's been around for going on 35 years. It's been around for a while and it's kind of like a take on the Nobel Prize. It's like the funny, fun one. But what's great about it is that like you're actually learning a ton from these kind of silly, like the research that kind of has a silly premise, but is legitimate research. And we do have an episode on the Ig Nobel Prize that came out, I think the end of our first year or maybe second year. I'm forgetting now. But anyways, I just wanted to interject in case a listener hadn't heard of the Ig Nobel Prize.
Yeah, and that's a good point because that's actually like kind of the story of this paper. This was a paper that was originally rejected by journals, but those scientists presented it as a poster at a conference, and researchers started forwarding it to each other, and it ended up leaving its mark on the field. Because basically, one of the things that this paper was showing is like, depending on how you filter your data, you can get results like this. So it kind of makes, it raises a lot of questions about like how to best analyze this data, because one of the challenges is like, yes, you can create a strong filter on your data that's going to say like, actually this dead salmon doesn't have any brain activity, but then you run the risk of actually losing data that's also real and that does reflect actual brain activity.
Right.
So, you know, this paper has had this long-standing effect on the field, including this more recent work that this article was about from 2020. So we're fast forwarding from 2012 to 2020, and scientists are still trying to figure out the reliability of fMRI. And there are these researchers who are working on a long-term study of 1,300 Duke undergrads looking for biomarkers that might explain why people process thoughts and emotions differently. And one of the ways that they're doing that is looking at brain scans, but they're also wondering how reliable these measures will be. And one of the central questions in this case is just how consistent can fMRI results be in an individual? Like if you measure them on one day on a task, are they like always going to have that same activity on a task if you measure them a week from now, 2 weeks from now? And that's really important because if you're using their brain activity as a biomarker, you need that biomarker to actually be consistent. Like it needs to be something that you would see reliably. So As part of a different study, some of the researchers, I think from this group, but also like just part of this team of researchers decided to see the reliability by looking at a number of different sources. So one of their sources was actually 56 papers that used fMRI data. They found that the correlation between multiple scans on a person were not that great. They also looked at something called the Human Connectome Project, specifically looking at tests of 7 brain function measures in 45 people, and then retests of those from 4 months later. And the correlation between those test results was pretty weak. Also looked at data from a study in New Zealand where people were put through a task-based fMRI twice to space 2 to 3 months apart. Again, very poor correlation. So it's just showing that like this individual measurement is like really not that reliable. But this also doesn't mean that fMRI data is like necessarily bad. It just means we have to be really thoughtful about how we use the data. For example, we might not be able to use it to say like specific things about a specific person's brain, but we might be able to average it out and understand like population level responses in general. Like we might be able to say things about the structure of the brain looking across a group of people. And there also might be ways to improve on fMRI, like maybe instead of collecting data, I think kind of like more on the order of minutes, maybe we should be collecting that data for longer on the order of hours, or using the data to see more about how parts of the brain are connected rather than just looking at what parts are active. So I think what it points to is that this field is still figuring out how to use fMRI data and what kind of like what's the best way to use this information and to think about it going forward.
That's really cool. I'm so glad you got into that because I knew about this study, but I actually kind of just knew that it existed and didn't think about the implications or realize that the field took it seriously enough to then be reevaluating how we use MRI data or like how we analyze and then interpret that data.
Right.
And that's a really important part of science.
Yeah. I think it's really good that the scientists working in the field aren't just taking it for granted that they can rely on their data points this way.
Yeah, absolutely.
Thank you to Cecilia, Madison, and to Caroline, Sam's sister, for submitting to Tiny Show and Tell Us, a bonus series from Tiny Matters created by the American Chemical Society and produced by Multitude.
You can send us an email to be featured in a future Tiny Show and Tell Us episode at tinymatters@acs.org, or you can fill out the form that's linked in the episode description. And we'll see you next time.